Okay, so question 29 and 31, we'll get it all done in one hit. So in a certain species of plant, the gene for stem colour assorts independently on the gene for plant height. The symbols for the alleles of the two genes are shown below. So we've got stem colour as B, big B for black and little b for red, T, big T for tall and little t for dwarf. Now we've got to use that information that we've given us, which is plant one, in terms of parents and what they actually are, to extract their genotype. So let's look at plant 1 first. I'm not even going to worry about plant 2 before I've done this. So plant 1 is a dwarf and a black stem. So we know from the get-go that in terms of plant 1 being a dwarf and having a black stem, that it's either going to be big B at the stem colour loci, and then it's obviously going to have to be little t. But we've got to work out which one that is. Plant 1 has a parent that has a red stem. Therefore, it's dash big little b. What we've extracted from this, though, is that if it's little b from this, then that must mean that plant 1 is heterozygous at that loci, which is big B, little b from this. So we know that the genotype for plant 1 must be big B, little t. Now, when we go to plant 2, we know it is tall and has a black stem. So from that, we need to look at this, and it says plant 2 has a parent that is a dwarf and red stem. Therefore, the plant 2 parent must be as follows: little b, little t, little d, which is homozygous recessive. Now, plant 2 is tall and black stem, which means it's dominant at both of those. So what we actually extract from that is that this must be heterozygous at both of those loci for those particular phenotypes, on the basis that this is a homozygous recessive parent. So therefore, this must be big B, big T, little t, for it to have actually gained that. So the 2, this is number 2, that is number 1. That's what we know so far. The question 29 asks us the number of different genotypes it's looking for, not phenotypes, genotypes, that would be expected in the offspring of a cross between plant 1 and plant 2 are what? So we're going to actually perform a cross here. So what we will do here, and I'll try and make it as clear as possible. I'm going to just go from there and split that up into the 2. So what we've got here is 2 possible combinations, which will give us BT or little bt. And so I'm going to put that up here and there. And then what I get from this is obviously we're going to get the 4 different combinations, which is big B, big T, big B, little b, big T, and little b, little t. And obviously there's a quicker way to do it, but I just want to be really elaborate with how I go, and then we can obviously do the genotypes from here. So big B, big T, little t, and I'm just going to keep writing them over and over. So we're back. I didn't really want to bore you with all the genotypes that I was drawing out, but what I want to actually put forward to you is that we've just performed that particular cross. And so what we've actually extracted from that are all the different genotypes. Now, the reason why I've crossed out these 2 is because they are exactly the same as this particular one over here and this particular one over here. And so when we count the number of genotypes we have 1, 2, 3, 4, 5, 6, which means our answer is C for 29. From that, we can actually go down now and do question 30, because it is looking for the number of different phenotypes that would be expected in the offspring of a cross between plant 1 and plant 2. So we've already performed the cross. Now all we really need to do is extract what are those phenotypes. So we know from here, we've got dominant at here, so that's going to obviously be black, and that is going to be tall. So we've got that first combination. We know we've got black dwarf, so that's obviously another different one. We know we've got black tall. I'm not going to repeat that one because we've got another one, so I'll tick that off. We've got black dwarf as well, so we've ticked that off again. And then we go to these ones, which was the same genotype. And then we know we've got this one, which is red tall and red dwarf. Obviously a lot easier way to look at it, but this is the way I've chosen to do it, and that gives us a total of 4, which is B. Then for the last one, which is question 31, we're looking at now plant 3, which is a new one that they've introduced, and we know it's tall and black stem, but we don't know if it's heterozygous or homozygous. The phenotypes of the parents of plant 3 are unknown. Thanks for being very helpful. And to determine the genotype of plant 3, so we want to look at the genotype of plant 3, it is best to cross it with a plant that is of what? Well, because we know they're both dominant at those particular alleles, then what I'd be thinking we want to do here is now cross it or do a test cross, and that's with a homozygous recessive individual. So a homozygous recessive individual will have a B, little b, little t, little t, which means it's red and dwarfed. And so therefore I'll look for it, and dwarf height and red stem color, that is D, shut up shop.